Welcome back to the deep dive. This time we are diving into a world that's so tiny you need a microscope to even see it.
Oh,
We're
wow.
talking about the cell membrane.
That's right.
You requested a deep dive into Chapter 7 of your biology textbook.
Excellent choice.
And that's what we're going to do,
Ugh.
unpack all that awesome science that's packed in those pages.
Absolutely. Cell membranes, you know, they might be small, but they're absolutely critical for life. They're like the gatekeepers,
I like that.
for the cell.
Yeah.
They control what goes in, what goes out, and they're involved in so many of the cell's most essential processes.
And to help us navigate this microscopic marvel, we have our expert in all things cellular.
Happy to be here.
I am so excited to learn about this. I have to admit, when I first read through the chapter, I was a little overwhelmed
I can imagine.
by all the details, all the terminology. We've got the fluid mosaic model and phospholipids, proteins, transport mechanisms. It's a lot to take in.
It definitely can be. But don't worry, we'll break it all down step by step. And by the end, I promise you'll have a much deeper appreciation for just how amazing cell membranes truly are.
That's what I like to hear.
Okay.
So the chapter kicks off with the fluid mosaic model. I remember reading that and thinking, okay, well, that sounds kind of cool, but what does it actually mean?
Yeah. It's a really elegant way of describing the structure of the cell membrane. Just imagine like a sea of lipids, kind of like a constantly moving ocean.
Okay.
And scattered throughout this sea are all sorts of proteins. Imagine those are like ships sailing around.
So it's not just a rigid wall then.
No, not at all.
It's more dynamic and constantly changing environment.
Exactly. And this fluidity is it's crucial for so many reasons. For one, it allows the membrane to kind of adjust to changing conditions. Think about temperature.
Oh, okay.
When it's cold, the membrane needs to stay fluid enough to function properly.
Right.
But when it's warm, it can't become too loose.
That makes sense. It's like how olive oil will become thicker in the fridge, but it flows really easily at room temperature.
That's a great analogy. The types of lipids in the membrane can actually change to adjust to different temperatures. It's like a built-in thermostat.
Wow, that's pretty cool.
Yeah.
So we have this sea of lipids and then these proteins that are floating around. What are the proteins up to?
Ah, yes. The proteins. Well, they are the real workhorses of the cell membrane. They do all sorts of jobs, like transporting molecules,
Yeah.
acting as enzymes, receiving signals from other cells, and they even help cells stick together.
Wait, so they're not just like floating around aimlessly. They have specific roles.
Absolutely. And the types of proteins that are present in a membrane, those determine the specific functions of that cell. It's like having a team of specialists, each with their own unique skill set.
So if we think back to that city analogy,
Hmm.
the proteins are like all the different businesses and services that make a city function, right?
Mm-hmm.
We've got the transportation workers, the chefs, the construction crews, the communication experts, each one contributing to the overall operation of the city.
Exactly. And just like in a city, there's a lot of communication and interaction going on between these different proteins. They work together to maintain the cell's internal environment, and they respond to changes in the external world.
Okay, that's starting to paint a clearer picture for me. But
Okay.
let's back up a bit and talk about those lipids that make up the foundation of the membrane. The chapter mentions something about a phospholipid bilayer. What's that all about?
Right. So the most common type of lipid in the membrane is a phospholipid, and these molecules have a unique structure. You see, they have a head that loves water, what we call hydrophilic,
Okay.
and then a tail that hates water, which we call hydrophobic.
Okay, so water-loving heads, water-fearing tails.
Mhm.
Where do they fit into this whole membrane picture?
Well, remember how the cell exists in a watery environment, both inside and outside?
Yeah.
So to accommodate this, the phospholipids arrange themselves in a really clever way. They form a double layer or a bilayer with their water-loving heads facing outwards towards the water
Right.
and their water-fearing tails tucked inwards away from the water.
So it's like a double-layered sandwich, right?
Yeah.
Where the bread represents those water-loving parts and then the filling represents the water-fearing parts.
That's a delicious way to visualize it.
And the whole sandwich is sitting in a pool of water.
That's right. And this unique arrangement creates a barrier that is selectively permeable, meaning that some things can pass through easily while others can't. And this selectivity is vital for the cell to function properly.
It's amazing how such a seemingly simple arrangement of molecules can create such a sophisticated system. I'm already starting to see why you said cell membranes are so remarkable.
And we've only just scratched the surface. But before we move on, are there any burning questions that you have about the fluid mosaic model or the phospholipid bilayer?
I think you've done a great job explaining those concepts so far. I'm particularly intrigued by this idea of selective permeability, though.
Yes.
How does the membrane decide what gets in and what stays out?
That's a great question, and it leads us perfectly into the next part of our deep dive, the fascinating world of membrane transport. So you're curious about how the cell membrane decides what gets in and what stays out.
Huh.
Well, that's a question that has fascinated scientists, for decades.
It's like the membrane is playing the role of a bouncer at a very exclusive club.
Oh, I like that.
Right. Some molecules get the VIP treatment and sail right in,
Yeah.
while others are left standing outside in the cold.
I like that analogy. And just like a good bouncer, the cell membrane has a set of rules that determines who gets in and who doesn't.
Okay.
And these rules, they're based on the size, the charge, and even the polarity of the molecule that's trying to cross.
So it's not just like a random free-for-all.
Not at all.
There's a method to the madness.
Absolutely. The membrane is incredibly selective, and this selectivity is crucial. It's really crucial for maintaining the cell's internal environment and ensuring that it can function properly. Now, the way things move across the membrane can be broadly categorized into two main types,
Okay.
passive transport and active transport.
Okay. Those terms ring a bell from the chapter.
Good.
Passive transport sounds like the easy route, no effort required.
Exactly. Passive transport is all about moving down the concentration gradient. Imagine you have a crowded room and you have a spacious one. People are naturally going to move to where there's more elbow room.
Right. It's like going with the flow.
Yes.
From where there's more of something to where there's less.
Yeah.
No need to push or shove, just ride the wave.
Perfect description. And this movement, it doesn't require any energy from the cell. It's a spontaneous process. It's driven by the natural tendency of molecules to kind of spread out. A classic example is oxygen entering cells for respiration. Oxygen is more concentrated outside of the cell.
Right.
So it naturally diffuses across the membrane to where it's needed inside.
Okay, that makes sense.
Yeah.
But not everything can just, waltz across the membrane.
Yeah.
Right. What about those molecules that need a little extra help?
That's where facilitated diffusion comes in. It's still passive transport, meaning that no energy is required from the cell.
Yeah.
But it involves special proteins that are embedded in the membrane that act like little helpers, facilitating the movement of certain molecules.
So these proteins are like the friendly ushers at the club, guiding certain guests to their designated areas.
Exactly. And there are different types of these helper proteins. Some are like channels, creating a direct pathway through the membrane for specific molecules to pass. Others are like carriers. They bind to a molecule and change shape to shuttle it across.
It's amazing how these tiny proteins are so specialized, each one designed for a specific task,
It is.
ensuring the right molecules get to where they need to go.
It's a remarkable example of how evolution has fine-tuned the cell membrane to be an incredibly efficient and selective barrier.
Yeah.
But sometimes cells need to move things against the natural flow, from low concentration to high concentration, and that requires a little more effort.
Okay, so that's where active transport comes in.
Right, you got it.
It's like swimming upstream. You need to put in some serious energy to make it happen.
Exactly. Active transport requires energy, usually in the form of ATP, which is like the cell's energy currency.
Right.
And this energy is used to power special protein pumps that move molecules against their concentration gradient.
The chapter gave the example of the sodium-potassium pump,
Yes.
which sounds like a real workhorse. Can you tell me more about that?
Oh, the sodium-potassium pump is a classic example of active transport. It's constantly working to pump sodium ions out of the cell and potassium ions in, even though there's already a higher concentration of sodium outside and potassium inside.
So it's constantly battling against the natural flow,
Yeah.
like trying to keep a beach ball submerged in a pool.
Exactly. And this constant pumping is essential for maintaining the right balance of ions inside and outside the cell, which is crucial for so many cellular processes, including nerve impulse transmission and muscle contraction.
Wow. It's mind-blowing to think that something as simple as moving ions across a membrane is so crucial for so many complex functions.
Yeah.
It really highlights the importance of active transport.
Absolutely. It's a fundamental process that allows cells to create and maintain an internal environment that is different from their surroundings, which is, essential for life as we know it. Now, we've talked about how small molecules move across the membrane, but what about larger molecules?
Right.
Like proteins or even whole bacteria? How do they get in and out?
That's a good question. I remember the chapter mentioned something about bulk transport.
Right. Bulk transport is how cells move really large molecules in and out. They use these things called vesicles. They're like tiny membrane-bound packages.
So instead of using doors or channels, the cell is using these little delivery trucks to move the big cargo in and out.
Yes, it is. And there are two main types of bulk transport.
Okay.
Exocytosis, where vesicles fuse with the membrane to release their contents outside the cell.
Right.
And endocytosis, where the membrane pinches inward to engulf substances and bring them inside.
Okay, so exocytosis is like sending out a delivery truck, and endocytosis is like receiving a package. Can you give me some real-world examples of these processes in action?
Of course, think about hormone secretion. Cells in your pancreas package insulin into vesicles and release them into your bloodstream via exocytosis, helping to regulate your blood sugar levels. Or think about how nerve cells communicate. They release neurotransmitters, which are chemical messengers, via exocytosis, allowing signals to travel throughout your nervous system.
So exocytosis is like the cell's way of communicating with the outside world.
Exactly.
What about endocytosis? What kind of packages are cells bringing in?
Endocytosis is, it's all about bringing in essential supplies or even engulfing whole cells. For example, white blood cells use a type of endocytosis called phagocytosis to engulf and destroy bacteria,
Oh, wow.
protecting your body from infection.
So our immune system relies on these tiny packages to fight off invaders. It's amazing to think about all the drama unfolding at the cellular level.
It is. And there are other types of endocytosis as well, like pinocytosis, where cells take in droplets of fluid, and receptor-mediated endocytosis, which is a highly specific process where certain molecules bind to receptors on the cell surface, and this triggers the formation of a vesicle.
Receptor-mediated endocytosis sounds very precise. Is that how cells take in specific nutrients or signals from their surroundings?
Exactly. It's a way for cells to be very selective about what they bring in, kind of like having a personal shopper for the cell. And this process is crucial for many important functions, including the uptake of cholesterol from your blood.
Wow, so all these different types of transport are happening constantly,
Yeah,
ensuring that cells get what they need and maintain a stable internal environment.
they are.
It's a real balancing act.
It is. And it all comes back to the remarkable properties of the cell membrane. Its fluidity, its selective permeability, and its ability to interact with a wide range of molecules and signals.
Okay, so we've covered a lot of ground about membrane transport.
We have.
We've explored passive and active transport, facilitated diffusion, and even bulk transport with exocytosis and endocytosis.
We have.
Is there anything else we need to know about how things move across this incredible barrier?
Well, we've covered the major players in membrane transport, but there's one more fascinating process we need to talk about, and that's osmosis.
Osmosis. I remember that word from the chapter. But I'll admit, it always makes me think of high school science labs and those shriveled potatoes.
I understand. Osmosis can seem a bit intimidating at first, but it's actually a very simple concept once you get the hang of it.
Okay, so remind me, what exactly is osmosis? It's just water moving around randomly?
Not exactly random. Osmosis is the movement of water across a selectively permeable membrane, like our cell membrane, friend.
Right.
From a region of high water concentration to a region of low water concentration. It's a type of diffusion, but specifically for water.
So water wants to go where there's less water. That seems a little counterintuitive.
It does seem a bit strange, right? But it makes more sense if you think about it in terms of solute concentration. Water moves to dilute the area with more stuff dissolved in it.
Okay.
Things like salts, sugars, proteins. So water flows from an area of low solute concentration to an area of high solute concentration, trying to even things out.
Okay, so it's not that water's attracted to where there's less water. It's more like it's trying to dilute the areas where there's more stuff dissolved.
Exactly. And this difference in solute concentration across a membrane, this creates what we call tonicity.
Okay.
And there are three main types of tonicity:
Okay.
isotonic, hypertonic, and hypotonic.
Those terms sound familiar, but refreshing my memory, what do they actually mean?
Sure. So isotonic means that the solute concentration is balanced inside and outside the cell.
Okay.
So there's no net movement of water. Think of it like equilibrium. Everyone's happy, no need to move.
Okay, isotonic, everything's balanced. What about hypertonic? That sounds a little more intense.
Hypertonic means that the solute concentration is higher outside the cell than inside.
Okay.
So water rushes out of the cell trying to dilute that outside environment, and the cell shrivels up.
Like those poor shriveled potatoes in the science lab.
Exactly.
So basically, the water is leaving the cell to go hang out with all the solutes outside. And what about hypotonic?
Hypotonic is the opposite. The solute concentration is lower outside the cell.
Oh.
So water rushes into the cell, trying to dilute the inside environment, and the cell swells up.
And if too much water rushes in, the cell could even burst, right? Like an overfilled water balloon.
Exactly. But cells have these clever ways to regulate their water content and prevent that from happening.
Yeah.
Plant cells, for example, they have rigid cell walls that prevent them from bursting in those hypotonic environments.
Right.
In fact, they actually thrive in hypotonic conditions because the water pressure, which we call turgor pressure, helps them stay firm and upright.
That's why wilted lettuce perks up in water. It's absorbing water and regaining that turgor pressure. Fascinating.
It is, and even single-celled organisms like paramecium, which don't have cell walls, have developed mechanisms to deal with osmotic pressure. They have these things called contractile vacuoles, which act like tiny pumps, constantly expelling excess water.
It's incredible how much effort goes into maintaining water balance at the cellular level.
It really underscores the importance of osmosis and the delicate balance that cells need to maintain. We've talked about how water moves, but remember, membranes are involved in transporting all sorts of molecules, right?
That's right. We've explored how small molecules move in and out, how large molecules get transported, and how water plays a crucial role in maintaining balance. What else is there to uncover about these amazing membranes?
Well, remember how we talked about those membrane proteins having all those specialized jobs?
Yeah.
Some of those proteins are involved in more than just transport.
Okay.
They play crucial roles in cell signaling and communication.
Oh, right. Like those receptor proteins we mentioned earlier.
Exactly.
They're like the cell's ears, listening for signals from the outside world.
Exactly. These receptors are incredibly specific. They bind only to certain molecules, like hormones or neurotransmitters. And when a molecule binds to its receptor, it triggers this cascade of events inside the cell, leading to a specific response.
So it's like a lock and key system. Only the right key can unlock that specific response.
A perfect analogy. And this communication system, it's essential for everything from coordinating our bodily functions to responding to environmental changes. Think about how your body regulates blood sugar levels.
Okay.
When your blood sugar rises, cells in your pancreas release insulin, which binds to receptors on other cells, signaling them to take up glucose from the blood.
So without these receptor proteins, our cells would be deaf to the world around them.
Exactly. And this communication system is just one example of the many complex and dynamic processes that occur at the cell membrane. But before we wrap up our deep dive, I want to highlight one more aspect: the amazing diversity of membrane structures.
Oh, yeah. I remember reading about all sorts of specialized structures, like junctions, infoldings, lipid rafts. It was a bit of a whirlwind.
It's a lot to take in, but it's fascinating how those basic principles of the fluid mosaic model can be adapted to create such a wide array of specialized structures.
Can you give me some examples of how these structures play unique roles in different cells?
Absolutely. Take membrane junctions, for instance. These are specialized structures that connect adjacent cells, allowing them to communicate and function as a coordinated unit.
So it's not just about individual cells doing their own thing. It's about them working together as part of a larger community.
Exactly. And there are different types of junctions, each with its own unique function. Tight junctions create a seal between cells, preventing leakage, like the lining of your intestines. Desmosomes act like rivets, anchoring cells together and providing strength, like in your skin. And gap junctions are like tiny tunnels connecting the cytoplasm of adjacent cells, allowing for direct communication.
It's amazing how these tiny junctions play such a big role in coordinating cellular behavior.
It really is. And then there are membrane enfoldings, which increase the surface area of the membrane, like adding shelves to a closet.
Right.
And this is especially important for cells involved in absorption, like those lining the small intestine, where microvilli, those finger-like projections, maximize the surface area for nutrient uptake.
So more surface area means more space for transport proteins, which means more efficient absorption.
Exactly. And even the lipid composition of the membrane can be specialized. Lipid rafts are regions of the membrane that are enriched with certain lipids and proteins, and these create these specialized zones for specific functions, like cell signaling.
It's like creating different neighborhoods within the city. each with its own unique character and purpose.
A great analogy. And all of this diversity just highlights the incredible adaptability of the cell membrane. It's not just a static barrier, it's a dynamic and versatile platform that plays a central role in, virtually every aspect of cellular life.
Wow, this deep dive has been truly eye-opening. I had no idea just how intricate and fascinating cell membranes are.
It's a testament to the beauty and complexity of life at the molecular level.
And it really underscores the importance of continuing to explore and understand these fundamental structures.
Absolutely, because the more we learn about cell membranes, the better equipped we'll be to address a wide range of challenges, from developing new treatments for diseases to engineering novel materials.
It's an exciting time to be studying biology, and the cell membrane is at the forefront of many groundbreaking discoveries. I can't wait to learn more.
Well, there you have it. A journey through the remarkable world of the cell membrane.
Huge thanks to our expert for being such a fantastic guide. I feel like I've gone from feeling totally overwhelmed to actually appreciating the complexity and elegance of these tiny structures.
It's been my pleasure. And remember, there's always more to explore. The world of the cell is full of wonders just waiting to be discovered.
Thanks for joining us on this deep dive into the fascinating world of cell membranes. We'll see you next time on The Deep Dive.